Okay, embryology. Embryology mini lecture. This is content from chapter 20 mainly. And we are starting from that fertilized egg. It became it underwent cleavage, and then it became a morula, and then a blastocyst. It developed an outer layer and was able to implant into the body of the uterus if everything went as planned. Now, I think, where did it go? I feel like we're skipping entire... Yeah, we'll go here and then we'll go back. Okay. So pre-embryo, from fertilization to 2 weeks. It's a zygote, a morula, and a blastocyst. It's an embryo until the end of week 8. It's a fetus from week 9 to 40. Now, again, this is an older chart from a different resource, and it did say that the fetal period was 8 to 40, which doesn't make sense because it's still an embryo in week 8. You can't be both. So I just went ahead and I changed that to a 9. And that's in accordance with some other resources that I found. So again, fetus from week 9 on. So the embryologic phase is a phase of differentiation. From here to here, we are differentiating. We are taking those stem cells, we are differentiating them until they can become fewer and fewer cell populations, until they become the final cell population that they're going to be. From that phase on, they may still undergo mitosis, but it's just more of that cell population. So that muscle cell becomes more muscle cells, that stomach cell becomes more stomach cells, that neuron becomes more neurons, right? So this is the fetal stage where we have a lot of mitosis and growth and development. So those are really distinct terms and those are really distinct phases. In fact, when you study embryology, you actually study through week 12 to complete differentiation. So I don't even know how I feel about that 9, but I did look it up and it is correct. Okie dokie, let's head on back. Some of this is a little different, weirdly done. We did that. One thing to point out is because this development is happening, this really, really early development from fertilization and cleavage into the morula and blastocyst, that is happening primarily in the uterine tube. There is such a thing as an ectopic pregnancy that could occur. So if we're able to develop that trophoblast layer while we're still in the uterine tube here, it's possible for implantation to occur out here. In rare cases, remember I mentioned that the ovary is not connected to the uterine tube. It's possible to ovulate into the peritoneum instead, and it's possible for those sperm cells to land out in the peritoneum. So it's also possible for all of this to occur out in the peritoneum, and then we could have implantation into the peritoneum, which is a rich blood source. So that pregnancy, it's not viable. It is absolutely non-viable, and it is very, very dangerous for the pregnant person to have an ectopic pregnancy. There was a politician once who said, well, can't you just move it into the uterus? And no, you cannot. We do not have those surgical techniques to move an ectopic pregnancy from the peritoneum or from the uterine tube into the uterus. That technique does not exist. Again, I'm not coming down on a side of the abortion argument. I am just giving you some scientific facts that let you know when politicians don't know what they're talking about. Okay, where were we? Okay, these are some weird pictures. I need a good picture to start describing overall embryonic development. Let's do gastrulation. Then come back to some of these. Yeah. Now, let's go back and forth between a couple of pictures. Let's go to this picture. So we have a blastocyst, and we have this outermost layer that's going to allow for implantation. These are the cells that become the embryo proper. Okay, it's right here. Now we're tracing those cells that become the embryo proper. We do have some fluid-filled spaces on either side of it, and of course that is distinct from, everything that's going on outside of it. So here's the actual, this is the part that's a person, or that's going to be an individual, right? It's not a person yet, it's some cells right now, wherever you land on that politically. At this point, the cells that will become a human are divided into two layers, and each of these has specialized. It's differentiated to a certain degree that we have an idea of where those cells are going to end up. We've developed, at this point, an ectoderm and an endoderm. Eventually, we're going to develop three layers: an ectoderm, a mesoderm, and an endoderm. And we actually have a really, really good idea of the destinies of these three different germ layers. This is the process known as gastrulation, developing all of the layers that are going to determine the general layout of the body. And it's always color-coded the exact same way. Okay, I did this a bit wrong. Let me fix this real quick. It's always color-coded the same way. The ectoderm is always in blue, and I might need more blue because my blue clay, I have some blue clay right here, but it's getting kind of gummy and weird. So you start out as basically a three-layer pancake. In your ectoderm, we always color code in blue. Your mesoderm, we always color code in red. I have a kind of pinkish color just because it's what I have in my clay. And then your endoderm is almost always color-coded in yellow. So if we're looking at it from the top, it's not exactly round. It's more of like that shape, that shape. So this is what you looked like before you were even, as you were just like implanting. That's what you were. Here you were. So there's fluid-filled spaces on either side of it. We're just focusing on these because this is the actual person part. The rest of this is extra. It's not extra, it's important to development. But these are the cells that actually become a human body. So in this top layer, it's not really top, there's no up or down here, we have the development of something known as the primitive streak. It's almost like a little line forms. And that's actually going to develop into your brain and spinal cord. So this outermost layer, the destiny of this outermost layer, this ectoderm, is mostly the integumentary system and the nervous system. So that's why it's color-coded in blue, is we already know the destiny of more or less those cells. Right along this midline here, you get the primitive streak. It develops something called a notochord. It's going to bunch up, and it's going to make a little tube, which I mentioned this is getting a little gumpy, so it's not really going to let me do anything that cool. But it bunches up into a tube at the top, and that tube is going to be your brain and spinal cord. Okay. Now, while shortly after this primitive streak develops the notochord, and that thing that's going to become your brain and spinal cord, you're going to have a certain amount of rolling. You're going to roll up like a ravioli. And this is what this is showing here. So you're going to go from being a flat pancake to a tube. You're going to do it laterally first. So what I mean is, this is going to be your head, this is going to be your butt. You were flattened out laterally, and so this is going to be your anterior body wall. So when you rolled up like that, you went from being a flat pancake, and you rolled in this way. And that's what you're looking at right now. So here you are rolling up side to side. Yep, they're still doing side to side. Here is that primitive streak is going in this axis. Here's that neural tube. So remember when we pinched up along this line, we made a little tube right here. So you have like a tube up here, and then you have these concentric circles for the rest of it. So that's why there's a tube here, and there's also going to be a tube here. They just didn't quite finish it. So that's part one. You develop into concentric circles with the integumentary system being this most superficial, but then you have the double tube with the neural tube. That's your brain and spinal cord. So all of that is ectoderm, integument, and brain, spinal cord, nervous system. You have your mesoderm. That's all of your musculature, your skeletal system, your blood, your heart, a couple of other systems. And then your yellow, your endoderm, that becomes your gut tube and associated structures. Your lungs also come from what was started out as your gut tube, but also your esophagus, large intestine, small intestine, stomach, anus, rectum, everything, as well as liver, pancreas. So all of your digestive organs come from your gut too. So you roll up this way. I think I've got some other sources for some of this. Yeah. How do we get to look like a fetus from this tube right here? We're also going to curve in cranially and caudally. The head and the butt are also going to fall in on themselves. Now I've run out of blue. It's not really covering anything particularly well. But as you can see, I've ended up with something that does not at all look like a fetus, but we'll pretend it does. It's more or less shaped like a little bean, right? So we got the right thing. We've got the skin on the outside like we're supposed to. We've got the gut tube on the inside like we're supposed to. Of course, this blue would entirely meet at the midline if I had enough blue. There, it's a little bit better. So that's how we get a person. I don't know if that worked or not. That was probably more confusing. Yeah. So again, we curl in laterally, then we curl in cranially and caudally. At the end of that, we start to look a little bit like fetus. There it is, curled cranially and caudally. Some of these pictures are just in a really weird order, and I apologize for that. I'll see if I can straighten them out, but I think it would be unfair to lecture one way and to post the slides in a different order. That might make it worse. Okay, so here we are, curled in cranially and caudally. This is in the 5th week, so this is still embryologic development. I personally, no scientist calls it this, but I personally call this the Loch Ness Monster phase. So we don't really look very human at this point, do we? That's why it's embryologic. It's still differentiating at this phase. We don't have our organ systems yet. We're still differentiating our organ systems. Once we get to the fetal period, we have all of the basic organ systems. None of them are operational. We have all of them. They need to grow a lot more before they become operational. Okay, this one's just way out of... Let's put this up here, because that one's just way out of where it needs to be. Okay, I'm going to do some cleanup. I'm going to go back and see if I missed anything, because I have my way of describing it. Did that. Mostly did that. I'm not doing a lot with the amnion, amniotic cavity, chorion. Technically, I'm supposed to talk about extraembryonic membranes, but I don't know that I want to talk about it a lot. Okay. So this is where that implantation initially occurred, and then there was a lot of growth and development, and eventually we developed a placenta, and the placenta is an important structure because it's the connection between the fetus's circulation and the parent's circulation. There is a blood placental barrier, so there's going to be a lot that's going to be blocked from crossing the placenta, which is important for things like blood type incompatibilities, for example, or preventing infections. Unfortunately, some viruses can and do get through, but ideally not too many. There's the remnant of the yolk sac if you do want to start making those connections. Let's try to track that yolk sac developing here, for example. Now the structure of the placenta, I'm going to quote the YouTuber Z Frank again. It's like a bloody pillow. It is like a bloody pillow. So you have all these capillary beds in the placenta, and of course there's going to be an umbilical vein and umbilical arteries. There's going to be two really important things happening across these capillary beds of the placenta. It's going to work for gas exchange as well as nutrient and waste exchange. So it's acting as the digestive system, the excretory system, and the respiratory system all in one. Because this is a fetus. It can't eat food. It can't use its lungs to breathe. All of those exchanges of waste products, nutrients, oxygen, carbon dioxide, they all have to happen across the placenta. So again, the process of the development of the placenta starts with implantation. We're going to create a pool of blood. The placenta seals off the pool, projects chorionic villi into the blood. The villi contain blood capillaries connected through the umbilical vessels to the fetus. So again, these are the maternal arteries and veins connecting to the, chorionic villi and the placenta, which is pretty cool, actually. Did that. Yeah, that was all in a weird position. We did that. Now, actually, like this level right here, we do have some more detail about what the destinies of these different layers are. Blue layer, ectoderm. Why is it this layer order? That's so stupid. Okay, this is the most superficial. This is middle. This is deep. So they put it in a weird order. Okay, ectoderm, most superficial. Of course, that's the skin. Also, the superficial part of your teeth, lens and cornea of your eye, lining your nasal cavity, superficial stuff. A couple of glands, your anterior pituitary gland. Remember that we do separate the anterior and posterior pituitary for a reason. Your neuroectoderm becomes your brain and spinal cord, as well as some motor neurons, some glial cells. Your neural crest is a special thing. Let me show you neural crest, just to show you more than anything. Embryologists are just really into neural crest cells. I mentioned the primitive streak and how you're going to have these two parts sort of bunch up on the side and create a neural tube. The stuff that was at the crests before they became a tube, that actually differentiates and goes elsewhere and becomes something called neural crest cells. And those go all frickin' over the place. They're your melanocytes in your skin, so skin pigment is determined by neural crest cells. So it's basically a modified brain cell. Think of this as like a nervous system structure that then goes all over your body and does all kinds of different weird things. So sensory neurons, part of your autonomic nervous system, your adrenal medulla. So again, it's a gland, but it's very related to your nervous system. Some of the bones of your face, your teeth, few skeletal muscles in the head. There's even parts of the great vessels of the heart that come from neural crest. It's not even on here. Why is this important? Why are these layers important? Why do I want you to know at least the very basic terms of what comes from what layer? It's interrelated to congenital disorders, disorders that are formed during pregnancy. Those are really significant. They can be really significant. They're very largely determined by the destinies of the cells. So when did the mutation occur that changed a cell population? What layer was it in? Was it an ectoderm cell that mutated and had a problem? If so, there might be a problem with the integument or nervous system. Was it a mesoderm problem? You might have problems with blood vessels, your heart, your muscles, your skeleton, your connective tissue. Was it your endoderm? Well, you might have problems with some part of your gut tube, maybe your lungs, maybe your liver, or maybe your pancreas, or one of these other things, right? And sometimes these congenital issues go with each other. So for example, if you have problems with neural crest cells, you might simultaneously have cleft palate and heart defects, because like I mentioned, there's some great vessels of the heart that actually come from neural crest cells as well. So these things tend to go together because these cell populations develop in the same places. So it's not a rote memorization task. It's something that prepares you for a lot of things that you're going to see down the road. There's that neural crest forming that I was telling you about. So here's the bunching. And then here it is turning into a tube, and some certain cells pinch off. And those are the neural crest cells that have a destiny all over. Did that. Did that. Did that. Did that. Okay, fetal circulation. Old picture, new picture. Now this is an adult view of the circulatory system with just some fetal circulation added to it, just for visualization purposes, which is why in some cases this is a little bit better. But I think this is more visible. So adults, this is typical adult. This is after birth. If you just remove the umbilicus completely, then you have an adult circulatory system here. You have the right side of your heart, you have the left side of your heart, they're closed off from each other. You have your pulmonary trunk to your lungs, you have your aorta to the rest of your body. That's what you're used to. I think you've seen this before. Now, in fetal development, you have some minor differences. Of course, the most important difference is a fetus is not breathing with the lungs. We are getting oxygenated blood through the umbilical cord. So that umbilical cord is bringing blood to actually the inferior vena cava. What? And that's taking oxygenated blood and it's mixed with deoxygenated blood in the vena cava and actually taking it to the right side of the heart. So, if we're taking it to the right side of the heart, we're not actually perfusing the lungs quite so much with it. We're not actually using the lungs to oxygenate. So we have to share that deoxygenated blood, or that mixed oxygenated and deoxygenated blood, with the left side of the heart somehow. We do so through two holes. A foramen ovale connects the right atrium to the left atrium. So this is actually going to be an opening, a foramen ovale. In an adult heart, that's a fossa ovalis. There's just a concavity there. It scars over after the baby is born and takes a breath. There's a second place to share this mixed blood between the two systems, and that is known as a ductus arteriosus. Also, after the baby is born, they take their first breath, the ductus arteriosus scars over. So two places where we mix oxygenated and deoxygenated blood, the foramen ovale and the ductus arteriosus. In the adult, this is the fossa ovalis, and this is the ligamentum arteriosum, right here. And all of those umbilical vessels degrade and go away. There you go. So the purpose of fetal circulation, again, is to bypass the respiratory system and mix oxygenated and deoxygenated blood. Cool, right? I think it's pretty cool. We've done placenta already. Process of birth. The last textbook I used referred to one of the functions of the uterus is to eject the baby during childbirth, or eject the fetus during childbirth. I always thought that was hilarious. I put a big red eject button, as though you just go, and just, and then you're done, which obviously is not the case. So at approximately 9 months, a fetus aged 9 months, the birthing process will begin. As you can see, we're going to need to have that cervix open up. That amniotic sac that was in white here has to rupture, releasing some amount of amniotic fluid. It is pretty typical, the illustration here is not bad, it's pretty typical for the bones of the cranium to sort of become misshapen while the baby goes through the birth canal. So this weird alien head baby shape is pretty typical for a vaginal birth. Of course, that's a pretty wide opening for a vagina and also a cervix. Very uncomfortable. It's an understatement. And of course, the part that the media usually skips over is that that placenta then has to separate from the uterine wall, and that placenta is actually going to be expelled as well as afterbirth, so to speak. Also pictured here, why pregnant women need to pee all of the time, and also rarely publicized fact that during childbirth it is very likely that you will poop on the table. It is very, very normal to poop during childbirth, but nobody ever talks about it. But there's why. And that's okay. The OB, the nurses in the room, they're going to wipe that poop off. They're going to go about their lives, and so are you. Various forms of testing during the prenatal period. Genetic testing, I had to edit. Another reason I really needed to update this is it movable? Yeah. We're not testing genetic defect testing. I don't think anybody calls it genetic defect testing. If your baby is born different, they're not defective. Don't say that. This has got to be like a really old slide. But, so genetic testing, we can sample the amniotic fluid, and we can do some biochemical studies and chromosome analysis, see if there are any atypical arrangements of their chromosomes, any atypical numbers, and give you some genetic counseling on what that is going to mean for you and for your fetus. And then we can also sample the chorionic villi, and we can do some more analysis from that. That's it. I think that's all I'm going to say about any of that. Let me know if you have any questions. Thank you for your time.